Chapter 4 covers cell structure. This is our list of study objectives that we're going to go through in this lecture. So cells were discovered in 1665 by Robert Hooke, but really not a lot of progress was made until the 1800s when the microscopes were getting better. Late 1700s, early 1800s. And in the years 1838 and 1839, two scientists working separately, Matthias Schleiden and Theodor Schwann, came up with what now is called the cell theory. And so you want to put a star next to cell theory. Cell theory has three parts, and you want to make sure that you know the three parts of cell theory. The first part is all organisms are composed of cells. That could be one cell for unicellular organisms. It can be many cells for multicellular organisms. But all organisms are composed of one or more cells. And cells are the smallest unit that are considered alive. So anything below that level in our hierarchy of organization, such as organelles or macromolecules, molecules or atoms, those things by themselves are not considered alive. So a cell is the smallest level on our hierarchy that can be considered alive. And number three, although these are in no particular order, cells come from other cells by cell division. So all cells that are on Earth today have a continuous line of ancestry all the way back to the first living cells, or you can say all cells today are descendants of the first cells that were on Earth 3.6 billion years ago. There is a rule in terms of cell size. There is a rule that's based on the ratio or the ratio of surface area to volume of a cell. So cells have to be small because they depend on diffusion to move materials into and out of the cell across that cell membrane, across that phospholipid bilayer that creates the barrier, which marks what's inside the cell and what's outside the cell. So that cell membrane has a surface area, and as cells get bigger, if you look at bigger and bigger cells, the volume increases much more rapidly than surface area. Mathematically, if you think about it, volume is units cubed and surface area is units squared. And so as the volume gets bigger, the surface area does not get as large. So we say that larger cells have less surface area per unit volume compared to smaller cells. Smaller cells have more surface area per unit volume. And so that surface area to per unit volume ratio has to be optimum. If cells get too big, it becomes less than optimum. So at a certain point of size, at a certain upper limit of size, cells cannot get bigger than that, or else they will have too much volume per unit surface area. So there's an upper limit on cell size based on that concept. So most cells are, 50 micrometers, maybe 100 micrometers, down to about 1 micrometer. So you're sort of in the 1 to 100 micrometer range for cell size. So in this slide, it says not too many cells are visible to the naked eye. They're microscopic. And I wrote most are less than 50 microns in diameter. This is the symbol for micrometer. This is a Greek letter mu. It looks like the letter U, but it has a little tail in front of it. And it's micrometer, micrometer. So most cells are in that, I said, 1 to 100 micrometer range. 50 micrometers sort of right in the center of that. But giving you a sense of the size of cells. Your eye cannot see these things, or if it can, it looks like the teeniest, tiniest speck. But even then, that would be a really large cell if you can see it with the naked eye. So most cells are microscopic. So we use magnification using a microscope to enlarge cells. So magnification makes the image larger, but we also need what we call resolution, which is clarity. We need the ability to see separate objects clearly separate in the image. and that's called resolution. All right. So except for vertebrate eggs, which can be seen with the unaided eye, most cells are microscopic in size. There's two types of microscopes that we use. One is called the light microscope and one's called an electron microscope. I would say two general types of microscopes. Within the light microscope category, there are some specialized versions, but light microscopes as a group use light. They pass light through a specimen, so there'll be a light source, usually a light bulb, and the light passes through the specimen up into the ocular, which is where you look. And that series of lenses combined with the light passing through the specimen creates an image. And the images are in color because the light is passing through the specimen. And so if there is color in the specimen, it shows up in the image. Electron microscopes, you do not look directly at the image. They use a beam of electrons, which is then passed through the specimen, and then it is creates an image on a screen or some kind of detector, and then you get an image from that indirectly. You don't look directly at the beam of electrons, but you can see the image as it's rendered on a screen after a beam of electrons is passed through the specimen. Electron microscopes are harder to use. You need more training to learn how to use them. You have to specially prepare the specimens, and they're very expensive. So electron microscopes run from $50,000 to $100,000. Light microscopes, actually I bought one recently for myself that was, I paid $150 for it. The ones that we use on campus are about $3,000 in price range, but much more inexpensive than an electron microscope. And an electron microscope always shows images in black and white. And so a lot of times they will artificially colorize electron microscope images. So say for a textbook, but that's an artist has selected a color. So the light microscope images LM, if it's on the image, LM, that means it was a picture taken through a light microscope. And if it says EM, then that's an image taken with an electron microscope. And any electron microscope images were originally black and white and then have been colorized by the artist or the publisher of the textbook. All right, so we're going to be thinking about cells, and we want to think about unity. Remember the unity of all of life. All cells, no matter what type of cell they are, have certain things in common. Number one, they all have genetic material. It is believed that RNA was the original genetic material, but currently cells on Earth have DNA as their chromosome or their genetic material. And so all cells have some kind of genetic material, DNA, and it can be broken up into pieces, which we call chromosomes. So some cells only have one chromosome for certain species. Bacteria usually each have one chromosome. That's pretty common. And then for other organisms, each cell may have more than one chromosome. But no matter what, the cells have some kind of DNA in them. Then they have something called cytoplasm. Cytoplasm is really a mixture of all the organelles and the cytosol, which is the cell fluid, the water-based material in the cell. But cytosol is an aqueous solution that all cells have. So cytoplasm refers to the entire contents of the cell other than the nucleus. And cytosol refers to just the liquid, the water-based part of that. So sometimes people use those words interchangeably, but there is a difference between them. Ribosomes are organelles that make proteins. So all cells have ribosomes. And all cells have a plasma membrane, which at its fundamental structure is a phospholipid bilayer that creates the boundary and defines what is inside the cell and what is outside the cell. So the plasma membrane, or sometimes we call it the cell membrane, is that barrier or that boundary for the cell, the official boundary of the cell. So we're going to look at the structures of prokaryotic cells pretty briefly. They're pretty simple in their structure, unless you really go into a lot of biochemical detail, which we're not going to do. But they're pretty simple. They have DNA, they have chromosome, but it's not surrounded by its own membrane. So we say it lacks a membrane-bound nucleus. The term membrane-bound means surrounded by a membrane. So the whole cell is membrane-bound, but then in some cells you're going to have internal compartments that are also surrounded by membrane. So the prokaryotic cells don't have their DNA surrounded by a separate membrane. So the DNA is just kind of wound up in the center of the cell or in one part of the cell. We call that the nucleoid region. Just outside the cell membrane, just outside the plasma membrane, is a layer of carbohydrate, which is the cell wall. The cell wall is outside of the plasma membrane. It's on the outer surface, so it's technically outside of the cell. We use the term extracellular to refer to anything that's outside of the cell. And the cell contains ribosomes. We said all cells contain ribosomes, so that's on this list. Ribosomes have what we call ribosomal RNA as a part of their structure. And there was a scientist, Carl Woese, who studied the ribosomal RNA of different organisms, and they found that there are very different types of RNAs that help us to classify organisms. And so by studying the ribosomal RNA, we've been able to come up with a really better way of classifying organisms. And then some species have flagella or other extensions that help them move. So that's locomotion. In this picture, they show flagella. It's kind of this tail, but other types of prokaryotic cells can have flagella on both ends of the cell, or you can have more than one, but in this picture, they just show one. And there's some other structures in this picture, but I'm not highlighting them. So a prokaryotic cell has everything that all cells have. So it has a DNA chromosome, genetic material. It has ribosomes, it has cytosol, it has a plasma membrane. And then additionally, most prokaryotes have this cell wall just on the outside surface of the plasma membrane, and some of them have these flagella. There are two domains that are prokaryotic. This should not be a surprise to you, archaea and bacteria. So remember, domain archaea and domain bacteria. And remember, those are all unicellular organisms in both of those domains, and they are prokaryotic. That means that there is no membrane surrounding the nucleus specifically. As far as cell wall goes, the bacterial cells have a cell wall made of a carbohydrate called peptidoglycan. So we talked about some carbohydrates in chapter 3. And in this case, we're talking about a type of carbohydrate called peptidoglycan. It has a very particular structure that we're not going to study or learn, but it is a carbohydrate. It falls into that category. Now, cell walls are common amongst most types of cells. The bacteria have them, the archaeans have them, the plants, the fungus, the protists. But even though all cell walls are made of carbohydrates, each group has a particular type of carbohydrate that makes its particular type of cell wall. And so in bacteria, it's called peptidoglycan, but in the archaeans and the plants and the fungus and the protists, it's made of a different carbohydrate in each case. Cell walls protect the cell, help it to keep its shape, and prevent it from taking up or losing too much water. Water is a key thing for life on Earth, right? Okay, so that is a good start to this chapter. And the next section, the next lecture part, will start with eukaryotic cells.